starting to become a unit to use for conservation at a subspecies level. So maybe this population looks healthy right now, but do their genetics look like they would be able to maintain their viability as environments change? So it's becoming more and more a topic of interest as climate change continues to happen, and people are starting to be concerned about the effects of climate and weather on animal populations. And in some cases, it's been seen to predict future risks that aren't exhibited phenotypically by animal groups, but mainly like inbreeding potential, or potential for inbreeding depression in the future. So we're going to move on to the study sites. So a lot of this work, I'd say almost all of this work that wasn't done in the lab, was done by Scott Smyers and colleagues of his. Scott is a biologist who happens to be here today. He works at Oxbow Associates. They're an environmental analysis and land use mitigation company. And Scott is, I've always called him a herpetologist because it seems to be the love where his passion lies. So he also is very involved with Wachusett Mountain, the Friends of Wachusett Mountain. So he kind of set up all this monitoring for these vernal pools. So there were 6 pools in particular where there was a lot of data. So the pools, the way they distribute, I started to think about treating them as 5 populations and 2 subpopulations. So 6 pools where 2 were so close together that they kind of are one population as opposed to 2, and I'll show you on the map. So down the bottom is Milford. So Milford's down here, that was a single pool. Acton's up here, and Westford's up here. There's 4.5 kilometers between those 2 ponds. And then this Princeton site is actually the Wachusett Mountain site, and there are 3 pools there. And I can show you. So you can see that, this is not big enough, sorry. There are 2 summit ponds, one at 600 meters elevation, the other one at 594. Those little numbers you can't see. They're really close to each other relative to other distances. And then this Matches Pond is at the base of the mountain, but it's about 1.7 kilometers away. So that's just all kind of relevant for thinking about how these populations distributed in their local adaptation.
Are all of the pools ephemeral? Do they dry up?
No, they're not.
None? Some?
No, some do. Scott could speak better to it for sure, but the Summit pool doesn't for sure. And I don't think the Matches one dries up completely.
So that would matter for predation, right?
Right. I will talk about that too.
Cool.
Thanks. Okay, so I'll bring that up again when it becomes relevant, but just to kind of start to get your placement on the map going. So I mentioned Wachusett Mountain. It's possible that many of you have been there. It's really popular for hiking. There's a steep slope there. It's good for beginners like me. And it's a unique environment for amphibian populations. It's a unique environment in general because it has the largest stand of old-growth forests in New England. Trees between 150 and 370 years old are there. An old-growth forest just means an undisturbed patch of forest for a really long amount of time. So it leads to a lot of unique environmental conditions. So up there, Scott has found that the topsoil is deeper and more moist in general. There are cooler average temperatures, probably because of the elevation. Greater annual rainfall. The hybrid period of the aquatic habitat is longer. Like I said, the Matches and Summit ponds don't dry up at all. And then there's greater predator abundance as well. And that stems from vernal pools drying up can't accommodate fish, because fish can't survive if it dries up completely. So the fact that these ponds are up there, and the fact that people like to dump their goldfish in the ponds when they don't want them anymore, leads to this predator situation that isn't common to all salamander populations. So the mountain salamanders have unique characteristics. Scott's also collected a lot of physiological data, like body size and length. There were also some studies done that were able to show that later reproductive maturity is kind of the norm in the montane populations versus the lowland populations. And then when I say larger, they're kind of heavier animals, and then when you measure a salamander, often you go, or any amphibian, you go snap to vent length, because the tail can be a variable length in case they lost part of it, for example. So you don't want to take the whole body length, but take the snap to vent length, or snap to curl, yeah, that's what it is. So when I say they're bigger, I mean they're longer and maybe a little heavier as well. So it kind of looks like there's some kind of there's definitely a local divergence between the montane populations and the lowland populations. So is that local adaptation? Would that be supported by distinct genetics in the community? So, like I mentioned, amphibians in general exist in these patchy distributions. They can become genetically distinct because of that. And then you have greater population genetic structure than you would in like a panmictic, like a fully mixed population of animals. So, this study just attempts to investigate the genetic distinctions and consistencies between the high and lowland salamander groups. It doesn't seek to the cause of the developmental differences or the evolutionary mechanisms behind that necessarily. We do talk about a few that could be at play, but we were just looking for a genetic correlation between the unique morphologies at the mountain. So to do that, we made a few predictions. Generally in ecology, the null hypothesis is that you're not going to see a pattern, if you don't see a pattern, there's no differences in allele frequencies. Means that these animals disperse with no geographic limit and they mate randomly, like they're panmictic, like I mentioned before. Typical null hypothesis. So we were thinking that if we saw a difference in allele frequencies, namely if they were similar in all nearby populations, but different from geographically distant sites, then that would mean there was limited dispersal structuring the population. So for example, going back to the map, if Westford and Acton were very similar, and then all the mountain populations were very similar, we were thinking that they were just limited by geography dispersal. And that was the only thing structuring their population structure. You could also say geographic distance predicts genetic distance. Or if there were similar allele frequencies in just some sets of nearby populations, that means that something else was maintaining the similarity. So if only some of our central Massachusetts populations are similar, we can kind of infer that there's some other mechanism going on that's creating their unique morphologies and unique genetics. And then another hypothesis we came up with was that genetic variation is more highly correlated with habitat type than with geographic distance. So it's not so much going to be so much a matter of how far away they are apart from each other as much as it is how similar are the environments in which they're living. So then we got to work. Collecting the animals, like I mentioned, was mostly done. Scott and probably a good team of volunteers over some years have been monitoring these anyway, so we took tissue samples from 1 year. Collecting the animals is kind of simple. You just set a minnow trap, which is just these inverted cones, into a wire cage, semi-submerged in a vernal pool that historically has had salamander breeding. And like I said, when they're in that congress and they're kind of in a daze, they'll go anywhere and they find their way into this minnow trap just on their movement alone, and they can't figure out how to get back out. They can't figure out how to go up and over, I guess. So it's pretty simple to catch them. Tissue collection can be done a number of ways. A lot of what was done this time was just toe snips. So, Rose, when you asked about marker capture, one way to mark a salamander is by doing a distinctive toe snip. So you're collecting or trimming away certain toes that you know from year to year what, like a stunted toe the next year means, like what year it was previously. Yeah, so salamanders have really high regenerative properties.
Right.
Dr. Braunhut is working with salamanders actually because of their regenerative properties. So it doesn't grow back so quickly that you can't tell that it had been lost at one point, but they do regenerate.
Thank you.
That I saw a hand. Sorry. So it's a good amount of tissue for extracting DNA, which is the next thing that occurs. And then that extracted DNA gets amplified with PCR at small microsat